Welcome back to Epigenetics Explained. For the next few episodes, I will be delving into the biology behind the various epigenetic mechanisms that drive changes in gene expression. These modifications generally involve alteration of the structure of chromatin. To recap, that is the combination of DNA and histones. Changes in the chromatin structure, as we saw in the previous episode in euchromatin and heterochromatin, affects its accessibility to transcription factors, which again are proteins that control the transcription of genes, and thus influence how easily genes can be expressed. Today, I will talk about the most widely researched epigenetic mechanism, DNA methylation. As the name suggests, DNA methylation involves the addition of a methyl group, which is a molecule that has 3 hydrogen atoms and 1 carbon atom, to DNA. You may be wondering, where are methyl groups added to in the DNA? Well, they typically target CpG dinucleotides. This is a pair of adjacent nucleotides in which a cytosine base precedes a guanine base in the 5' to 3' direction. Hence CpG, which stands for cytosine phosphate guanine. CpG islands, which are regions rich in CpG dinucleotides, can be found in around 60% of gene promoters. Remember, this is the start of a gene. The reason why DNA methylation targets gene promoters more than any other location in the genome, which is our entire DNA blueprint, is that CpG sites across the genome are typically already methylated, while promoters are usually unmethylated. Now, what does this all mean and why is it important? Enzymes called DNA methyltransferases, or DNMTs for short, transport methyl groups onto the 5' of a cytosine base. This converts the cytosine to 5-methylcytosine, which changes the structure of the molecule itself but not the base sequence. After the methyl group binds to the cytosine, a transcription co-repressor complex, which is a group of various repressive proteins, prevents transcription factors from binding to the gene, thus preventing gene expression and essentially turning the gene off. So, DNA methylation generally represses gene transcription and expression. It is often involved in X chromosome inactivation, a process in which one of the 2 X chromosomes in females becomes inactivated, as well as aging and carcinogenesis, which is the development of cancer. DNA methylation usually is a relatively stable epigenetic mechanism. Long-term transcriptional silencing can result from DNA methylation during development. However, DNA methylation isn't irreversible. Demethylation is the removal of methyl groups from DNA through either active or passive means. Active demethylation involves the addition of a hydroxyl group onto 5-methylcytosine, which is the methylated version of cytosine, by 10-11 translocation enzymes, or TET enzymes for short. This converts it to 5-hydroxymethylcytosine, which makes it possible to turn it back into unmethylated cytosine. Passive demethylation is merely the loss of 5-methylcytosine through repeated rounds of DNA replication, which happens when there is a lack of DNMTs. Speaking of DNMTs, I want to take a step back to these trusty enzymes. There are 3 main types: DNMT1, DNMT3A, and DNMT3B. DNMT1 is considered a maintenance methyltransferase, and this is because it usually targets hemimethylated DNA. That is, when just one of the DNA strands is properly methylated, rather than completely unmethylated DNA. Therefore, it is most important during cell division, when the two DNA strands split to create daughter DNA, and one of the daughter DNA strands already carries the existing methylation patterns. This is how methylation patterns are lost through DNA replication without DNMT1. On the other hand, DNMT3A and DNMT3B are de novo methyltransferases. De novo roughly translates to anew from Latin and refers to DNMT3A and DNMT3B's ability to methylate unmethylated DNA as well as hemimethylated DNA. These DNMTs are most important during differentiation of stem cells and development. The reason why you are learning this is to understand how these enzymes become compromised in diseases such as cancer, which we'll be learning about in a future episode. One final note which will be useful for understanding how DNA methylation is implicated in carcinogenesis. Hypomethylation is the lack of or decrease in DNA methylation, while hypermethylation is the excess of or increase in DNA methylation. I hope you've enjoyed the third episode of Epigenetics Explained, and see you next time for our next epigenetic mechanism, histone modifications.